Polytechnic National Institute for doing his master's and PhD as well in the area of immunology. Then during his PhD, he did a research year at the Oxford University, and he has been working since 2016 at the Rutgers University, working in the immunology department. And I'm very happy to have you here, Nuriban.
Thank you, Henio. Okay, so I hope everyone can see my screen and hear me well. Okay, well, thank you again to the organizers for inviting me. It's very exciting to present my work. We published this a few months ago in PNAS. So our lab is interested in the transcriptional regulation of tissue-resident lymphocytes and other compartments too. So I'm going to present what we found in pregnancy progression related to IL-33 from the maternal side regulating pregnancy progression in the mouse model. So as a brief introduction, so for the past 20 years, we've known that every time we have more and more information showing that the maternal immune compartment, they orchestrate pregnancy progression in mammals. In both human and mice, there's work related to this, and as Kenichiro just showed, there's plenty of information, and it's very interesting how the immune cell compartment is also nurturing and protecting the host, in this case, in the maternal-fetal interface. So the immune cells here, they are different flavors. We have the myeloid compartment, macrophages, and dendritic cells, but also the lymphoid compartment. NK cells, I mentioned that our work is very focused on NK cell work, which are very important in this compartment. You wouldn't think about an NK cell as it's a natural killer cell, that it's related to host defense, but they're also very nurturing in this environment. Apart from that, we have other lymphocytes like Tregs or CD8 T cells, but also innate lymphocytes of the type 2. So overall, the function of these immune cells throughout pregnancy is to regulate and modulate that everything goes smoothly, so you have a positive pregnancy outcome in mammals. So since pregnancy is such an important process for the reproduction of the species, so it has to be very well kept in line, and no disruptions have to be occurring whatsoever, so you have a successful outcome. And as Kenichiro gave an excellent introduction about pregnancy progression, so I will not dwell too much on that. But in our case, so as I mentioned, any disruption in these signaling pathways that are related to pregnancy homeostasis, they will have poor pregnancy outcomes. And there's clinical data showing that one particular, very interestingly, I realized that this today's seminar is related to IL-1 family of proteins, which actually one of them is IL-33. It's also related to the IL-1 family that Kenichiro just talked about. So we know that there's, for the past 10 years, there are several papers of clinical data showing that perturbations of these signaling pathway that are associated with IL-33, they have associations with poor pregnancy outcomes in mammals. So the clinical data in humans, they are different papers showing a dysregulated expression or abnormal levels of IL-33 or its receptor that are observed in patients with preeclampsia, recurrent pregnancy loss, preterm labor, among other conditions. For example, there's a positive association of SNPs in the IL-33 locus associated with recurrent pregnancy loss in humans. Also, more recent work has shown that IL-33 can act on the residual B cells in late pregnancy, and this is associated with a protection against preterm labor. So as you can see, there's human data showing that IL-33 signaling pathway is critical for correct pregnancy progression. While in the case of rodents, there are data showing that IL-33 is involved in a similar, as Kenichiro just talked about, the LPS model. So there's a model in rats that IL-33 deficient rats, dams, are protected from complication induced by LPS in late pregnancy. In our case, our group has previously shown that in mice, IL-33 is expressed in the uterus, and interestingly, this expression is modulated by the estrous cycle in the mice. And we know that if the expression of this pattern of expression is maintained at the onset of pregnancy. But what is IL-33? So I've been talking about a little bit of that. So as an introduction about IL-33, as I previously mentioned, so it's from the IL-1 family of cytokine. So do we have that in common? It's a pleiotropic cytokine, which means that it's acting on a different wide range of cells and exerts different functions depending on the cell type. But something that is very particular about this cytokine is that the difference between other cytokines that we know, like IL-1 or IL-6 or any other pro-inflammatory cytokines, is that this cytokine has a nuclear location. So it's expressed and is kept in the nucleus of the cell. And interestingly, it was discovered that it was released during cell death. So that's why it was called an alarmin. So it gives this alarm sign of tissue damage or cell death. So most of the classical data showing about IL-33 function is being associated with tissue damage in the context of infection or any other tissue damage or cell death. However, more recent data by different groups have shown that it can be secreted actively via poorly endorsed mechanism. So it's still not quite clear. Originally, it was thought that 33 should be produced by dying cells or dead cells, but now we know that it's cells that don't have to be dying or dead to be actively secreting this cytokine, but it's still not clear yet what's the exact mechanism for that. Another interesting thing about IL-33 is that the cells that express it in very high levels are what we're calling structural cell types, which are not your classical immune cells that you will think of. These involve cells in the epithelium, in the stromal compartment, and also in the endothelium compartment. It's not exclusive of these cells, but they are very highly expressed. It's very enriched in these cell subsets, but also the immune cells can produce it in certain conditions. So the receptor for IL-33 is a dimer comprised of these two subunits. ST2 is the name of the exclusive chain that is recognizing IL-33, but they also need the other monomer is the accessory protein that is common to other IL-1 type of cytokines. So this is the signaling pathway. So these IL-33 signals on a broad range of immune cells, both of the myeloid and the lymphoid compartment. And this has been shown in a lot of different tissues, like the adipose tissue, or the muscle, the gut, the lung. So there's a lot of this acts in a lot of different tissues. So it's known to be an initiator of type 2 immune responses. And that's because the signaling pathway that IL-33 activates involves that adaptor protein MyD88, and finishes with the activation of NF-kappa B and the production of different pro-inflammatory cytokines, classically. And these type 2 immune responses are classically associated with either parasites or allergies, or in our case, we are very interested in the tissue remodeling compartment. So in other tissues or in other conditions, this is associated with fibrosis or other types of negative outcomes, but we are interested in the tissue remodeling that occurs in pregnancy. So this would lead us to our hypothesis, that is that based on the human data and the mouse data that we know, is that IL-33 signaling will promote uterine tissue remodeling that supports pregnancy progression and for that we're going to use a mouse model. So Kenichiro gave us a very nice introduction about mouse pregnancy, so I'll just do a quick recap of that. So the mouse pregnancy lasts around 19 to 20 days, and we can classify it by at least in 3 different subsets or times, like as early pregnancy between day 5 to day 8, that is characterized by the implantation of the embryo at day 4, and this triggers a process known as decidualization, which here at 7.5 involves the differentiation of the stromal cells surrounding the embryo. So they differentiate to give more a nurturing environment and to protect the embryo, and to also there's a lot of blood vessels vascularization occurring. So that has to occur, so the embryo starts to develop, and that needs to be it's optimal in its development. Then decidualization should be done by day 7. So for the next process to start, that is called placentation, that starts around 8, and it should be done by day 10 when the placenta is finally formed, And it's about the period that we know as mid-pregnancy. So here I'm showing you on the right from this paper, it's 12.5, how a mouse pregnancy looks around day 12. And now by day 12, we have already formed a placenta with the deciduum, which is most of the immune cell compartment. It's the maternal side is occurring, and then we have your fetus. That after the placenta is formed around day 10, then the fetus starts to grow for the next week or so that involves late pregnancy, which is between day 15 to day 19 when labor occurs. So based on this model, so we wanted to know if first IL-33 was still expressed in pregnancy in mice. So as I mentioned, we were interested in early pregnancy first. And so we used a reporter mouse. So this mouse has a citrine cassette inserted in the IL-33 locus. So it will report this fluorescent protein whenever IL-33 was expressed. It will be fluorescent and citrine. So we wanted to focus at 7.5 when the association was completed in early pregnancy. So we wanted to know whether IL-33 was expressed in the uterus. And we found it by different techniques. So in panel A here, you can see it's some immunofluorescent analysis of IL-33 expression cells. So in green, you will see the citrine reporter that is expressing IL-33. In C31, in red is the marker of the vasculature, and that is the nucleus. So in this case, you can see that the IL-33, the green signal is associated in this line, in the lining of the uterus that is called the myometrium, or the muscle layer of the compartment, where most of the IL-33 positive cells are expressed. But we also detected some IL-33 expression in the decidua, which is this differentiated endometrial tissue surrounding the embryo here at the center in the lowercase e. But the IL-33 expressing cells in the decidua were actually located in this region in the rectangle, white rectangle, that is called the mesometrial pole. So it's this region where most of the spiral arteries that are coming from the uterus are sitting. So if we do a zoom in this region here at the bottom part, we can see some green cell types that are co-expressing C31, which again is a marker for blood vessels. So we compare then that IL-33 expressed in situ in the pregnant uterus at 7.5. And we also use flow cytometry in B and C to compare the expression of IL-33 in the pregnant uterus. So interestingly, the take-home message from B and C is that IL-33 is expressed in the pregnant uterus starting at day 5 when we looked at, and it's still expressed by day 9. But very interestingly, it's very enriched IL-33 expression in the myometrium compartment here in black, compared to the decidua, where it's a very lower expression compared to the myometrium. So this also was confirmed by Western blot analysis in D, when you can see that the full-length protein of IL-33 is expressed in both compartments, but the mature form is seems to be enriched in the decidua at 7.5. So the take-home message from this slide is that IL-33 is actually expressed in the uterus in mice at early pregnancy. So then we know that it's expressed in this compartment. We wanted to know what was the identity of the cells expressing IL-33 in mice. So for that, we use a single cell sequence analysis. So we sorted and sequenced IL-33 expressing cells from our reported mice and analyzed the expression and the identity of IL-33 expressing cells. So we split it by analyzing the myometrium at 7.5 and the decidua. Excuse me. So in the myometrium, the IL-33 expressing cells, we could identify 4 different clusters based on the gene expression analysis. And interestingly, we found that the cluster included myofibroblasts that are expressing this ACTA2 gene. We also found stromal or fibroblast type cells that are expressing PCGFR alpha or podoplanin here in F, but also smaller clusters of epithelial cells that are keratin 8 expressing cells, and also vascular cells, which are the endothelial compartment. We confirm our findings of RNA-seq analysis with flow cytometry analysis, and we can see, as you can see, that around more than 80% of the cells that express C3 in the pregnant uterus from 5 to 9.5 are these stromal cells. So these results are in line with other papers showing that the stromal or the structural cell types are very enriched of IL-33 expression in other organs. In the case of the decidua, if you just to remind you, we have very few cells expressing IL-33, and most of them are in the mesometrial pole where the vasculature, the spar arteries are located. But our IL-33 expression cells, we can find again 3 different clusters that are vasculature and also stromal cells here, clusters 2 and 3. Interestingly, this stroma could be subdivided by the decidualized stroma, which expressing these genes, HOX11, WOIN4, and HAN2, which are associated with this differentiation of the stromal compartment induced in early pregnancy, but also the podoplania. And again, by flow cytometry analysis, we could confirm the identity of these cells between 5 and 9.5. And again, we saw that most of the cells are stromal cell types. However, very interestingly, at 7.5, the vascular CD31 positive cells are also expressing it. That coincides with what we showed previously in mesometrial pole expression. So the message of this slide is that we identified that myometrial fibroblasts and decidual stromal endothelial cells are the main IL-33 cell types in the uterus in early pregnancy. So now that we established this, we wanted to know what's the effect of the not having IL-33 on the maternal side in pregnancy progression. So we use IL-33 deficient mice, female mice, that were mated with wild-type mice, and we compared that also with wild-type females mated with wild-type males. So the first thing we looked at was, again, at 5.5, when it's right after this, the embryo was implanted, and deserialization starts, which is a very important event because that involves a lot of tissue remodeling that has to be occurring for the pregnancy to progress. So by using confocal analysis, here we could determine by cytokeratin 8 staining the implantation chamber length. So you can see here in a wild-type female, the white line, it's the distance of implantation chamber length in the epithelial compartment. So the embryo is in the center. And that's the distance that we are measuring here in the epithelial compartment. When we compare that with knockout females at 5.5, we noticed that the implantation chamber length was smaller compared to the wild type. So we know that there's maybe a defect then of the pregnancy on the tissue remodeling at early pregnancy at 5.5. If we looked at 2 days later, when the sterilization is completed, we also noticed that the implantation side weight was decreased in the knockout females compared to the wild-type females. And very interestingly too, at 7.5, if we measure genes associated with decidualization that should be completed by this time point, we notice that the knockout females have decreased expression of these WIN4 and FKBP5, which are genes associated with decidualization in mice. So overall, we can say that the maternal deficiency of IL-33, it's impairing the uterine decidualization and this tissue remodeling that is necessary for optimal pregnancy progression. So that's on the mother side. But we want to know, okay, so what's the effect that this has on the embryo compartment? So we did the same thing. We look at the embryo development starting at 5.5. And here again, using confocal microscopy, we could analyze the volume of the embryo. So we can see here in this white circle oval, we can measure the volume of the embryo at 5.5, surrounded here by the epithelium and cytokeratin in green. So as here are 2 representative images, as you can see, the embryos are smaller in the knockout females compared to the wild types. And when we measure this by volume, again, significantly decrease the size of the embryo volume at 5.5 in the knockout females. If we look 2 days later at 7.5, there's a staging of the embryo development that should occur properly at 7.5. So this is characterized by the presence of these particular features. that should occur in wild-type mice, as you can see. The neural plate is NP, should be occurring by this time point. But when we compare how was the embryo development in the knockout females compared to the wild types, we notice that most of the knockout females, they have their embryos were situated in a delayed developmental stage compared to the wild types. Most of them were in the pre-stage compared to the neural plate phenotype, which suggests that the embryo development at 7.5 was delayed or affected by the maternal deficiency of IL-33. And more strikingly, if we look at them 5 days later, at 12.5, once the placenta has been formed and the fetus starts to grow, we notice that here's some representative images that the knockout females, they have a higher increase of frequency of resorptions, which means that implantation sites that didn't develop properly and they are not compared to the other leader maize that you can see here. There's the placenta and the embryo inside. But in the knockout mice, we found an increased frequency of resorptions. So, and then, so the conclusion of this of this work in this part is that maternal IL-33 is required for the optimal development of embryos in the early pregnancy. It has this impact at mid-pregnancy. Another important feature that occurs during deserialization in pregnancy, in early pregnancy, is there has to be a vascularization, proper vascularization, so the nutrients can go to the embryo very readily. So the residual vascularization that is occurring also by day 7.5. So we wanted to know whether IL-33 deficiency might be affecting the residual vasculature in early pregnancy. So to do this, we analyzed the expression of CD31, which I mentioned previously. It's a marker for vascular cells. And we also analyzed alpha SMA, smooth muscle actin, which marks the mural cells in the pregnant uterus. So here's a representative example of how CD31 staining looks in 7.5. So all of this in green is a capillary network of different vessels. As you can see, they are, depending on the site that you're located in the uterus, they are bigger caliber vessels in the mesometrial pole here at the top, and smaller vessels in the anti-mesometrial region at the bottom of this picture. So we measure the CD31 density of these vessels in different regions of the implantation site. And what we found is that in the case of the central region here surrounding the embryo, or the anti-mesometrial region at the bottom of this picture, we didn't see a difference in CD31 density. However, we noticed an increase in CD31 signal at the mesometrial pole, which is coincidentally where we saw IL-33 expressing vascular cells at 7.5 with our reported mouse. So we compare then wild type versus knockout dams. We notice this increase in the CD31 density, which means there's an abnormal decidual vasculature in early pregnancy. Interestingly, we didn't see differences in the density of alpha SMA, which suggests that at this point, there is no defect in the spiral arteries at 7.5. So another process that is involved in surrounding vasculature remodeling that occurs 2 days later at 9.5 is called the spiral artery remodeling. And this process is regulated, it occurs by 9.5, and it involves here our H&E staining representative images of here these are spiral arteries. So here you can see the round arteries. And they usually, the remodeling involves that the mural cells compartment that you hope you can see here, they should decrease in size, which involves a higher volume blood flow. So that should occur to appropriate blood flow and spiral artery remodeling. So we compare this remodeling between the wild type and the knockout females, and we found that very interestingly, the knockout females at 9.5, they have a higher vessel area, as you can see here in the arrowhead, and also the lumen area. So their spire arteries were not properly remodeled. As you can see, the wall thickness was increased in the embryos from, in the implantation sites from knockout females compared to the wild types. Although the ratio was conserved, but we saw that the knockout females, they have thicker walls. And this process is mediated by immune cells, such as mast cells, regulatory T cells, and NK cells. So which means that the deficiency of this cytokine on the maternal side is associated with a poor spire artery remodeling at 9.5. So this is the results at 9.5, but in the end, we wanted to know what is the outcome in case of the pregnancy progression in mice when the females don't have IL-33 on the maternal side. So for that, we analyzed our mice very late in pregnancy, almost 1 day before labor at the 18.5. And we wanted to know how was the pregnancy outcomes at this time point. So we are analyzing the uterus weight, but more importantly, the number of viable fetuses. And very interestingly, when we have a female knockout mated with wild-type males, we found that they have a decreased number of viable fetuses, which was coincidentally with an increased number of resorptions in the knockout females compared to wild-type males. However, this was not the case when we did the opposite mating, which was wild-type females and knockout males, because then they have resorptions, but they were not the ratio was not as high as when the females lack IL-33 on the maternal side. More interestingly too, we noticed that the fetal weight of the pups coming from the knockout dams, it was decreased compared to the weight of the wild type by wild type or the knockout by wild type matings. And more interestingly too, out of these smaller pups overall, if we analyze the frequency of small for gestational age, which ACJ stands for, the frequency of them occurring at 18.5, it was very interesting to see that the pups coming from knockout dams, they were 25 out of 74 of them were classified as small for gestational, which means they are below the 10th percentile of the wild type by wild type matings. So again, lacking IL-33 on the maternal side critically affects the development of the pups at late pregnancy. Not only early pregnancy, I have shown previously here, but also at late pregnancy. And that also affects the placental growth because their diameters were smaller compared to the wild type by wild type matings. So overall, we have shown so far that the deficiency of maternal IL-33 has critical effects on the pregnancy progression, affecting, both deserialization, vasculature remodeling, but also overall pregnancy progression and viable pups. But as I mentioned early in the introduction, IL-33 acts is coming from stromal endothelial or vascular cells, but it acts on immune cells. So in order to know which immune cells were affected or were responding to IL-33 in the pregnant uterus, we use an unbiased approach, which involves single-cell RNA-seq analysis. And what we did at early pregnancy at 7.5 is to sort CD45 positive, which involves immune cells, from the myometrium and the decidua of wild-type females, and then doing a reclustering, analyzing the cells that express the ST2 receptor, which the name of the gene is IL-1 RL1. We wanted to know which cell types were expressing the receptor for IL-33 in early pregnancy. So we found these 4 clusters, and based on their gene expression analysis, we could identify different populations of immune cells that are expressing ST2 in early pregnancy. So cluster 1 involved CDC2s, which are these immune cells associated with type 2 immune responses, antigen-presenting cells, dendritic cells. Cluster 2 have ILC2 or T cell gene signature. Cluster 3 had a signature involved in macrophages, which are poised to the M2 or alternative activated type of response. And finally, cluster 4 involved mast cells. So all of these immune cells are associated with type 2 immune responses, which I mentioned early in the introduction. They are associated with tissue remodeling or these defense in type 2 responses. When we compare also by immune flow cytometry, the expression of these ST2 positive cells, we could confirm by flow cytometry that indeed the ILC2s were most of the cells that were expressing ST2 among lymphocytes in the myometrium. And these results are in line with other tissues showing that ST2 is expressed by these type of immune cells. In the case of the decidua, which again is the tissue surrounding the embryo at 7.5, when we saw it on CD45, we again found 3 different clusters. And with similar results as the myometrium, cluster 1 involved a signature associated with CDC2s or M2 macrophages, which are associated with tissue remodeling or anti-inflammatory responses. Cluster 2 included regulatory T cells, which are very important for maintaining tolerance and in the pregnant uterus. And finally, cluster 3 again included mast cells. So then again, the difference between here, the decidua, and the myometrium, the uterus lining, is that in the decidua, most of the ST2 positive lymphocytes are regulatory T cells compared to immune cells. Compared to the myometrium. So now that we established which cell types can respond to IL-33 in the pregnant uterus, we wanted to inquire whether the deficiency of IL-33 in the maternal side will affect the function of these immune cells in the pregnant uterus. So one way to assess the immune function of immune cells in these type 2 responses is to measure the cytokine production by these cell types. So here I'm showing you intracellular cytokine staining for 2 main cytokines that are associated with type 2 immune responses, classical immune responses, which are IL-5 and IL-13. And here I'm showing you the expression of these 2 cytokines by ILC2s, these innate lymphocytes. And here are representative plots. And then the quantification compare the ratio between wild types versus knockout females. And interestingly, we found that the knockout females, they have lower expression of these type 2 associated cytokines, IL-5 and IL-13, which is in line with other findings from other groups and other tissues where the deficiency of IL-33 is associated with these impaired type 2 immune responses by these cytokines. When we, if you remember, we also found that ST2 was expressed by T cells. And in this case, when we analyze conventional T cells, which are FOXP3 negative, and again, assess IL-13 expression by intracellular cytokine staining, we again found a decreased expression of IL-13 in the knockout dams compared to the wild types. So IL-5 and IL-13 are upstream of other type 2 immune responses that are associated with these type 2 responses. One of them is the recruitment of the viability of eosinophils, which are these granulocytes that are present. They're actually highly present in the uterus. And interestingly, we found that at 2 time points, either 7.5 or 9.5, we noticed that the knockout females, they have less eosinophils in their tissue compared to the wild type females, which is again in line with this impairment of the IL-5 and IL-13 production by ILC2s and T cells. Another cell type associated with these type 2 cytokines that are responding directly or indirectly to ST2 signaling are macrophages of the M2 type or alternative activated macrophages. So one way to phenotype these macrophages is by their expression of arginase 1, which is this enzyme associated with M2 macrophages. So when we quantified the number of macrophages expressing arginase 1 in both the decidua or the myometrium, they were decreased at E7.5, but also at 9.5. So overall, we could show that the deficiency of maternal IL-33 has an impact on the type 2 immune responses associated at this early pregnancy in early pregnancy. So this finding coincides with the defects that we saw in deserialization, but also in the vascular remodeling at 7.5. And finally, So the summary of this work is that the pregnant IL-33 knockout dams, they exhibit defects in uterine tissue remodeling and deserialization in early pregnancy. We know that maternal deficiency of IL-33 has a delay early embryo development and increased resorptions. I've shown you that the knockout dams have an abnormal decidual vasculature and spar artery remodeling in early pregnancy. We also identified the sources of cellular IL-33, which are myometrial fibroblasts and decidual stromal endothelial cells as the main types. We know that the receptor is expressed by immune cells associated with type 2 immune responses in the uterus. And finally, that these defects in the tissue and the vascular remodeling are coincidental with an impaired immune response, type 2 immune response in knockout dams in early pregnancy. And finally, I would like to acknowledge the people involved in this work, my PI, Amy Bolio, in my lab, our collaborator, Nataki Douglas, also at Rutgers, New Jersey Medical School, but also Dr. Goss at Rutgers too, and our collaborators at Michigan State with Ripla Aurora, our NJMS core, and of course the funding provided for this work. And with that, I'm happy to take any questions. Thank you.
Thank you very much, Nuriban. I think it was a really great talk, and I think we learned a lot. At least I really enjoyed your talk. So there are several questions in the chat, so I think I can proceed to read them. Eric asked what is the signal that induces IL-33 expression during this few days of pregnancy?
So that's a very good question. So interestingly, IL-33 is expressed there. So it's constitutively expressed. So what will trigger its release, which is if it's actually released, I would think so. It's still not clear yet. We know other people from Arimolovsky's group, they have shown that there are these niches in near the vascular part of different organs, like the lung, that they interact. They're very close to each other, ILC2s, Tregs, dendritic cells, but also these stromal cells. So probably they are together, and there's some signal that it's making IL-33 to be secreted. But my guess would be it has to be very tightly controlled, because otherwise, I mean, IL-33 is a very potent cytokine, and as I mentioned earlier, it's associated mostly with tissue damage. And of course, in pregnancy, you don't want that. It's the least you want, damage or cell death. So my thought would be that there's going to be a very controlled secretion of this cytokine, but we know that it's important. So yeah, it will be very interesting to know what is triggering its secretion, not like the expression, but the secretion during pregnancy.
Okay, thank you, Nuriban. There's a question from Angel David. I don't know if you want to turn on your mic, or I could proceed to read. He asked if fat tissue increased or decreased IL-33 expression.
So the work from Diane Mathis, and I remember other labs, mostly they have focused on the adipose Tregs. I think they have shown that the stromal cells in the fat, they express 33. And they have also other groups from Lydia Lynch, they have shown that it's also associated with interleukin-17 responses and gamma delta T cells. So we know that it's there, and there is associated with like thermogenesis or other like obesity. Also is being associated that obese mice, they have more IL-33, if I'm not mistaken. So yeah, it's associated with that.
And Christopher asked and I think there was a question also I had in mind, if because you should only show like the immune compartment that can respond to IL-33, but if there are any epithelial cells that can respond to IL-33 in the placenta?
That's a very good question. We haven't analyzed the non-immune compartment, but there's evidence that ST2 can be expressed in other non-immune cells. What it does there, I'm not sure. mostly, I think what we know on ST2 is on the immune cell compartment, what it does. But I guess, yeah, we want to know, we have continued this work, and now we have conditional deletion of ST2 in different cell types, and we want to actually address that part on the immune compartment and the non-immune cells.
You know, and finally, Edgar Guzman says, like, if there's any relation between maternal microbiota and the modulation of IL-33 expression during pregnancy.
That's a good question too. We haven't addressed that part about the microbiome in pregnancy. I don't know the answer to that because I'm trying to remember other papers that have analyzed IL-33 in the vaginal tract, which is of course not the same as the uterus. I'm trying to remember if there's anything known about that. Yeah, I can't remember, so I don't know the answer to that question.
So I guess I will ask my final question, and there are different groups that have shown something about inflammation, like infection during pregnancy have a repercussion of I think Nardi's group have already done that, but I was wondering if you could expose the mites like to a systemic type 2 immunity. Could that have a defect directly on the fetus? Like, I don't know, I'm thinking about it, and maybe, for example, the nipple infection, that you could control the days of where the nipple is in the organism, that could, you know, have a implication.
That's also a very good question too. I'm trying to remember. I think there are data showing, or at least anecdotal, or I've heard about it, that parasitic infections, they have some problems with pregnancy. Like I think schistosoma. There's some evidence about that. I would hypothesize that since STAT3 is associated with that immune response, that you would, of course, you have a decreased response. So since we know that it's important in pregnancy, like to have a proper pregnancy progression, if the parasite is there, maybe it could be even worse outcome, I would think. It would be an interesting experiment to do. That would be one option. Another one I was thinking was when you said like systemic type 2, like an allergy. That could also be another one. I'm not sure it's been done like that type of response. But I would at least anticipate that the knockout females, they will probably couldn't respond properly to the parasite, maybe for that. But in the case of the allergy, I'm not sure if they will have any problem. Yeah, but it will be interesting to do.
Thank you very much, Nuriban.
Sorry, can I ask you?
Yeah, sure.
Question?
Oh, yes, yes,
sorry. So, if I understood correctly, your mating system is Y-type male and knockout females, correct?
Correct.
So, what happens if you use both knockout mating?
We haven't done that experiment with knockout. Because we were focusing on the maternal side. We wanted like to leave out any issue from the maternal side, the paternal allele in the pup, at least when the pups are in the fetal compartment. We know by anecdotal evidence that the knockout mice, and also there's a paper showing that the ST2 knockout mice that are made like ST2 knockout mice, they have maybe less pups. But there's no, like, I don't remember there's any formal evidence that they what may happen there, but I would expect that they may have maybe even a higher increase in resorption. So like in this defect, I would anticipate that. Okay. Because we know we have the data here in the one I show in the other mating when the sire is knockout. There's a defect in that, so that tells us that there's a component on the paternal side and in the pup, but we haven't analyzed that part on the offspring side.
Okay, thank you. Because we have IL-33 and ST2 knockout, but we don't see the rest pups. But we didn't analyze them like in detail as you did, like you did. So we just made them, and then just they are mating well, but they have a pups well. But the slight reduce of number couldn't detect it by our mating system. So maybe it's interesting. And then the other question is that, did you see the implantation side of IL-5 knockout or IL-13 knockout mice? Because the last outcome is the expression of IL-5 and IL-13 in ILCs. And then also it's very interesting that you saw the neutrophil, the eosinophil count of the uterus.
Yeah. So the first question about 5 and 13, we haven't analyzed those mice, like the knockout mice, but I would also anticipate, and coming back to your first question, I think that's a very interesting thing, because that's what we thought too. I mean, as long as the mice breathe regularly well, I don't think anyone has looked at carefully like how the pregnancy is affected, because as long as you have pups in the end, you don't really care. But that's why we're like proposing here, that it's not that simple, because there's probably, I mean, yeah, you have pups in the end, because pregnancy is such an important, like crucial phenomenon that has to occur, that you have to like have redundant or something to make it work. So having said that, that's why I don't think people have seen before that they maybe have a problem or not, like doing that proper experiment. And that's why we haven't done that also in 4 and in 5 or 13 knockouts. But I would expect something similar as in here. We are thinking of probably doing a STAT6 knockout mice to confirm this. I would expect that. I know there are some groups working on MIT88 knockout mice, which possibly maybe have some problems too. It's a different, I mean, it's another cytokine, as you know. It's another other different activation with MIT88, but I would expect that the IL-5 or the 13, they may have some defects. Again, I'm not sure how dramatic will be compared to the 33 knockout mice, but I would expect to see some problems also in that part. And when in the with the eosinophils, what was your question again? Sorry.
Oh, last question is just a comment, because eosinophil, no one was analyzing the number of eosinophils in the uterus in detail, right? So
yeah,
then
it's a
and then sorry, we don't know the real role of eosinophils in the uterus. So I'm really interested in that.
Yeah, no, that definitely. That was actually like a comment from the reviewers, that they were like, we should like hypothesize like what are the eosinophils doing there. And yeah, I completely agree with you. I'm sure you're familiar with the papers showing the IL-5 knockout mice. They seem to be having regular pops, but they never went to see pregnancy, like how if there was any difference when they don't have 5 or the one that don't have eotaxin. Yeah, I think in the case of eos, I'm also fascinated by them. They're there all the time, as you probably know now. But yeah, they less of them. They start what they're doing. I've read that they might be associated with the tissue remodeling per se. Like they have like the metalloproteinases and some like extracellular matrix remodeling. So I'm sure there must be they were doing something related to that. Because that's where we're like trying to focus, like the remodeling. So pregnancy is such a dynamic process that there has to be occurring all of these. So yeah, I agree with you with the EOS. It's very fascinating and intriguing cell type.
Yeah, thank you for that. That's a really cool study. Thank you so much.
Thanks. Thank you.
Thanks, Nuriban. Thank you, Genichiro. And thanks everyone for joining today. And so you can follow our social networks as well. And you can also, if you have any interesting topic you wanna, or a particular topic you wanna join us with organizing a seminar, we will help you through that. And thank you once again, Genichiro and Nuriban, and congratulations on your work. Bye-bye.
Thank you, Enio.
Thank you so much.
Nice to meet you.
Thank you, Nuriban.
Bye.
Nice to meet you.
Bye.